Thank you very much. Good afternoon. Some of you still awake. The sandwiches were good, right? So what I'm going to try and do over the next, how long have I got? 15 minutes? I have very strict instructions to talk for 12.3 minutes or something. Something around that is to keep you awake, I hope, but also to give you an insight into what the Medicines Discovery Catapult is here for. And that's really quite easy, actually, because all I have to do is ask you to get out your phones, don't turn them on, and look at them. And what do you see? It's you. Because that is actually what the Medicines Discovery Catapult is really here for. So now, the name's on the agenda, so I don't count that. But before today, how many people in this room had heard of the Medicines Discovery Catapult? Oh, actually, that's better than I normally get. Good. Well, hopefully, within the next 12.3 minutes, you will get an insight into the Medicines Discovery Catapult, and I hopefully will have 100% of hands up. Those people who don't put their hands up, those sandwiches were really good, and therefore you've gone to sleep. I hope that that's not the case. So I have a pointer here, and it works. Right, I was always taught that if in danger, have one slide that if you were stark buck naked, you could talk to for 15 minutes. This is it. I'm clothed. I wore a tie today, particularly and only for the microphone, and that I'm scared of my own buttons. So apologies for being slightly overdressed for the event. This is what the Medicines Discovery Catapult is all about. It is here as part of the overall commitment of the UK government to the life sciences, as a delivery agent of the life sciences industrial strategy, targeted at the small to medium-sized companies, those companies who have a very still, small voice that's often drowned out by big pharma, to help them, to help translational academics and technologists, and in our case, medical research charities, gang up on the problem that we have in medicines discovery, identify the barriers that exist, collaborate to break those barriers down, to produce new products and services that we can push out to the CRO community and start new companies with that will help raise the tide for all. One of my colleagues, Mark Samuels, sitting at the back, who runs our engagement office within the Catapult, and I do constantly tell him he has a very, very easy job, which I have to do over the phone because he's a big guy. The reason I think that he has an easy job is he has to listen to one thing, which is that. Whenever you're in a room, you'll hear that, oh, somebody really needs to, and you go, okay. That's a problem that no one person can solve, that everyone knows about, that if it's solved, helps everybody. Somebody really needs to. And there are a number of somebody really needs to problems in the medicines discovery field. Over the last year, we produced some scholarship which we published in January called The State of the Discovery Nation. I have a few copies up the front, and we'll make sure that hopefully the organisers can send around the link to it. What that did, for the first time in about 10 years, was go deep into the biotech community and ask them what they really needed to help them be successful. And there were 10 or 12 things on there, but uppermost in their minds were, help me get better preclinical models, help me collaborate in a new way, help me manage and deal with the tidal wave of information that all of this stuff produces, and therefore let me attract money. It's not give me money, it's let me attract money through the strength of my academic endeavour and my sweat and the ability to jump over a few barriers that are there for all. And you'll see those reflected in our actions. Everyone's seen the chevron diagram for about 4,000 years, so I've taken the chevrons out just to keep you awake. The most important thing, however, about this diagram, ladies and gentlemen, is this bit here. For many years, and 30-something years since I've been in this wonderful profession, we have had a massive patient-free zone in discovery. And that cannot be right. We've slept-walked our way to doing this for decades, where we have taken a publication and generated an unmet patient need from it. It's wrong. We have to start with the patient. with consented patient data and consented patient samples and the voice of the patient and the view of the patient at the beginning, and then we might have a chance of being more predictable as we get to this stage here, which is the first time that we've generally involved a patient, i.e. when we really need one. And that's really not where we should be. But more to the point, that linear model doesn't reflect reality, and it certainly doesn't reflect the future. We used to take a publication, find something out, say that we'd validated it in vitro. Really? In vitro? With the sort of tools and techniques we've got, we think we've validated a clinical target in a tube. Wrong. We hadn't. We'd associated it nicely, and that's good, but we need to recognize that. We need to start doing stratified discovery, not getting all the way to a phase 3 and then doing some sort of post hoc idea about how beautifully we stratified this medicine. Start it early on disease subtypes. That means we need to think in a different way. We need to add to the complexity of our preclinical models. Move away from reductionist models. I'm not sure that many of my cells right now are growing in a complete homogeneous single cell layer on plastic. Very few of them, I hope very few of them are right now. But that is the way in which we test new potential drug candidates. Why is that? Why do we expect them to work, let alone why do we get worried when they fail? If our models are more predictive and correlate just by 0.1 greater towards the norm, but by 0.1, it's not much, right? If we improve by 0.1, Scannell et al. Have written that they believe that's equivalent to over 100 times brute force effort. So we can carry on, if we like, screening in plastic, in single cells, in nicely transformed, chemically derived, patient-unlike cells, and hope by brute force we will find the medicines of tomorrow. Or we can think a bit more humanized, complex, difficult, but predictive. And these are some of the missions of the Medicines Discovery Catapult. And as we move around this list, and you can all read, you will see a stratified approach coming in at an earlier and earlier stage, and target validation, ladies and gentlemen, is done again in the patient, not in a publication. All of this produces a mass of data. And unless we start to link it properly, none of the machine learning, none of the AI systems have a hope of working. They have been around for a long time. We all know that. They've been made effective because we've linked data together. So let's start in the right place by linking data more effectively, providing pools and lakes of data, connecting omics, outcomes, and biology, and chemistry where relevant, so that the AI algorithms can be given a chance to really learn, really be trained on a proper real-world data set. So the Medicines Discovery Catapult, we've been around about a year or something, based in Alderley Park in Cheshire, which somebody said, where in Oxford are you from? Actually, Cheshire in Oxford. I mean, I'm sure that at some point your organisation will take over to the whole of the country, but for the moment we are in Cheshire, in the former site occupied by AstraZeneca, who went to the other place as a relocation. And it's really important to note, for those of you who have not been there or not been there recently, how vibrant this has become in around about a year. And the sort of numbers of companies that you're forming here are being formed up in the northwest. And they are living here. They are populated by and run by industry-savvy people who want to keep doing stuff, who are well-trained, and we've got facilities there for them. Wow, what an advantage. They have not got the multi-millions that are sitting around here, but they share the same ambition as you, and they have great facilities, so it's important to keep watch on them. In order to address these barriers, then, we have put together 4 essential capabilities in discovery, science and technology to industrialize the models of tomorrow. That is 3D, organ on a chip, complex co-cultures, and the bioanalytics that wraps around them. Because it's all very well to grow a co-culture and it works once every other Tuesday if you turn left and hum, but that does not make an industrially robust, disseminatable, usable system. So somebody really needs to do that. Looking at the bioanalytics that wraps around them, New techniques in complex mass spec, tissue imaging and so on are necessary to really understand the complexity and deal with the complexity of those models. But again, they need to be joined with them, they need to be tested, and they need to be robust. So we are doing that in the safety space and as well as the efficacy space. So complex mass spectrometry, organ on a chip, those types of technologies are how we are starting off our life. We're also dealing with microbubbles drug delivery systems there, doing some industrialization of those systems, because some of the great drugs that you might find don't need to be once-a-day oral. I know that's anathema to those of us who were back in pharma in the day. We would never have been allowed to stand up and wear our little badge without saying once-a-day oral, but it's not necessary. If you can target your drug to site of action, release it locally, cargo it locally, reduce tox, reduce cost of care, then you're in business. So delivery systems, formulation systems, those things which often SMEs leave till the last minute, quite understandably, because it might go wrong and it might be difficult, bring those in early because that adds real value to your product. So think about those things. In informatics, we have a capability run by Professor John Overington, formerly of Benevolent AI and the chap who wrote Kemble. That is all about joining data together, making big data interoperable to allow your algorithms to be tried, tested, industrialized on really high-quality global data, to be able to look at a target and say, what does the world know about this? Where is the patent estate? Where's the chemistry that's gone wrong on this? Who owns the chemistry that's gone wrong on this? If you have a tool, which bits of it don't you own? All of these questions de-risk your study, de-risk your company, increase the value for investors who are looking to invest in you. In virtual R&D, and I'll come on to that in a minute, virtual R&D is the practice of outsourced research and development. It's been mastered by Big Pharma, it's been used by SMEs. But there's a difference between using and having mastery of something, as I find out when I play my son at squash. Patient sample and patient data access does put the patient at the beginning of discovery. It's a vital necessity for the future of medicines. But 75% of today's SMEs, people like you, end up going abroad for access to consented patient samples and patient data. And that, frankly, ladies and gentlemen, is a travesty. With the reserves that we have here of consented patient samples and patient data, we should be on Olympus, and we're not. We're down in the foothills of the Chilterns somewhere. We need to improve that. Okay? It's a bad statistic, but it's also a global problem. No one's got this right yet, but we are aiming through our brokerage to give you better access, find out where samples are that you need, help you through the contracting process to get access to those samples, help you use them if you don't have the capabilities to use them, and then add quality labeling to those samples so no one ever has to go through a 9 to 12 month wait to get a set of samples that are unusable. This, again, is wrong. So that acts as a bit of a turbo for medicines discovery, and it enables SMEs to come and have access to collaborate around, to develop new tools and technologies that are going to help everybody succeed, yourselves included. But there's something that this turbo engine also drives, and this is the new form of consortium. A consortium that is disease-based, has a portfolio of drug candidates within it, cornerstoned by a research charity, and what we call drug discovery syndicates, which Mark Samuels at the back. Mark, put your hand up, please, so everybody can see you. Mark, who is actually also a mountaineer, which is why he prefers the top of the room. Mark's group is running this syndicate operation. This is really important, and it's really important because a medical charity, unlike venture capital, and unlike big pharma, never falls out of love with its disease. In a 13.5-year process, which is idea to market, a pharma can have 4 or 5 really strategic changes in what diseases it cares about. A 5-year VC fund, unlikely, but a 10-year VC fund can change. Putting together drug discovery syndicates gives assets the best chance of being incubated in an environment where everybody is chasing the same disease, everyone's following similar pathways, and where the research charity can bring the voice of the patient, the key opinion leaders, the patients, and numerous upstream biology resources to bear on the problem. This is ganging up on the problem, ladies and gentlemen, which we must do. There are over 100 disciplines between idea of a medicine and putting it first into a patient. Let's not all kid ourselves that we all have those skills at hand. We don't. We need to gang up on the problem. The syndicate approach is the best way of doing it, we believe. And what it does is it allows assets to pop out at the time when it's right for a really good management team to take them and push them hard. So we're not starting companies that are science-rich but asset-poor. We're starting companies with a really good balance of science wealth and asset wealth. Really important construct. So please do talk to Mark or myself about that later on. As I said, we are focusing down in our discovery science areas on these areas of discovery to help some of these tools and technologies come to the fore. And as we are helping those technologies be developed, we're asking SMEs, come, bring your assets, try them out, see if these add value to you, see if they move your product from one step to the next step more effectively. So join us in that endeavour. We're going to be making calls out there, asking for you to come to us with your assets, your waifs, your strays, and help you to prove them in some of these more modern methods. And the same with virtual R&D. 40% of the UK's SME companies have got less than 5 people. So that's about 95 skills short. Many of those skills, capabilities, assets, tools, technologies, assays are available somewhere in this nation. The issue, ladies and gentlemen, in this country is not that we haven't got the assets, it's your access to them. So the job in virtual R&D is to provide pharma-class outsourcing networking for you to answer these 3 basic things. What experiments do you need to do, 2-3 experiments do you need to do that are critical to making your asset industry consumable or financeable? Secondly, where in the country can those things best get done? They could be in CROs, could be in specialised academia or elsewhere. And then help in making that complex, outsourced, multi-party discovery happen. That's what virtual R&D is all about. So we're building that platform right now and building that national network of academic and industrial drug discoverers who can provide pop-up advisory teams for an afternoon to tell you those things. So virtual R&D is a service to the SME community that will help you get Pfizer class in an afternoon. But those are very active things that we are doing. These are some of the things which may be regarded as passive, but actually also, we hope, very helpful. The UK is full of acronyms. We love them because it makes us feel as though we know something that other people don't. In reality, what it does is it just confuses people. There are huge numbers of organisations who are built to help you. Not just in Oxford, I'm afraid, but across the country. And the Catapult is a single USB to those organisations. You need to know where some of these folks are, what they do, how they can help. Call us, because we are here to help you get the best value from them, and we are here for them to help them help you. Because let's also remember, in a publicly funded environment, and most of these are publicly funded, they will somewhere in their grant renewals have, I have helped some SMEs. Now, in order to help SMEs, they actually have to meet some of you. And that's one of our jobs too. So don't be shy about coming to us and saying, I don't know anyone in NIHR, and I'm not even quite sure what the acronym stands for. Well, as Mark will tell you, when you climb the mountain to meet him, the NIHR is about £1.2 billion a year of public money going into healthcare research. They are here for you. The BRC is here for you, right? So for those of you who need additional help in linking into those first-in-man trials and other things, come and talk to us. And so what's in it for? I was asked what's in it to express what's in it for Oxford. So I have to do the what's in it for Oxford. Apart from what Nick is talking about in terms of this ITAC consortium, which Chaz and Chris Schofield and others are helping put together to address diseases of ageing, a national consortium, but you know, conceived and born here. We also want to make sure that for the SMEs in your community, you get access to assets, to our capabilities, to our interventions, and ultimately you take away these outcomes. It's really important to us, and we believe to the fabric of the UK biotech environment, that you take these outcomes home. And by doing that, you will attract the funding that you deserve. And with that, I'll finish. Thank you very much for your attention.